Hi, I'm Pete. I'm one of the scientists at Tufts, although at the moment, because of the coronavirus, I'm actually a scientist in my kitchen. What I'm studying, both in the lab and to a lesser extent in the kitchen, is the metabolism of the bacteria that cause Lyme disease. And we're doing that because we think it might help us to design new drugs. The particular thing I'm interested in is the way that the bacteria kind of acquire all the nutrients they need to grow. And it's interesting because the bacteria is a parasite. It can only really survive in a host, whether that's a tick or a mouse or a person. And it needs to be around a host because it's evolved over time to basically become very lazy. It's lost a lot of the genes it needs to make the nutrients it needs to grow. So what it does is it just steals them instead. And that happens quite a lot with parasites. And also someone I used to date in college. So we sometimes visualise metabolic activity using these kind of subway style maps where the dots are chemicals and the lines are kind of pathways to synthesize or break down specific nutrients that the organism needs to grow. So sometimes these are quite complicated. This is the human metabolic map. So you've got, like, in here is the circle for making energy. Up in that corner is for the bit that handles caffeine. Down in the bottom is the place where cholesterol is made. Now obviously the human metabolic map is very complicated. We can also look at simpler organisms, and what we can do is we can look at species, so you can find out what the differences are between species, what makes species unique. For instance, this is yeast, and we can isolate from yeast the specific pathway that makes alcohol out of sugar, which is handy. So one thing we can do is to compare the map for Borrelia, the Lyme disease bacteria, with maps of other similar bacteria, and what we find is that Borrelia's is much smaller, and that's because they have a much smaller genome, so they only have 1,200 genes, where a lot of other bacteria maybe have 4,000 or 5,000. So the upshot of this is that the metabolic map doesn't look quite so much like the complicated kind of London Underground map, say. It looks a bit more like the Boston Metro, which only has 4 lines. Let's say you're in London and you want to go from Liverpool Street to Hammersmith. You can use the yellow line. If you don't like the yellow line, you can use the pink line. If the pink line's bust, you could go red and then blue. Or if you've just got a lot of time to kill, you could do something stupid like this. And you've got a lot of options because it's a very complicated map. Where there's a lot of complexity, you've got a lot of flexibility in terms of different routes to meet the same objective. If you compare that to, say, Boston's Metro, which is much simpler, much smaller, say you want to go from the Tufts building in Chinatown, where our lab is, down to the Sam Adams at Stony Brook, you use the orange line. If the orange line's broken, tough. There's no other way to get there. You just have to stay in the lab and you can't get to the pub. So one of the things we're doing in the lab is working to try and understand the metabolism of Borrelia better, which should let us draw a kind of more complete and more accurate version of these metabolic maps. And we're doing that because it helps to design new drugs. And Borrelia in particular we might be able to target because it's got this very reduced metabolism. We think that if we can design drugs that block specific pathways, Borrelia won't have the sort of metabolic flexibility to cope with any new challenges we throw at it. And anything we do throw, any pathway we do block, should be lethal and should be a good drug treatment for Lyme disease. Because if we do manage to kill Lyme disease, then I can probably go to the pub